Hello, biologists. We're going to continue talking about osmosis and diffusion. And this time we're going to ask ourselves how many molecules, or many molecules, even a single atom ions can diffuse easily through the cell membrane. How do they get into and out of cells if they cannot easily diffuse through the cell membrane? So that's really important. Let's think about how you would get through a cell membrane if you can't diffuse through. Remember, the outside of a cell membrane is polar. The polar heads of those phospholipids make the outside polar and the inside polar. Kind of the middle of this sort of sandwich of cell membrane is nonpolar. So a lot of things have a hard time passing through here because it's polar and nonpolar at the same time. Let's look at what we can do to help something get through. Because water is polar. Most molecules need a pore, in other words, a hole in the cell membrane that lets them go through. An aqua, or pore, helps water get through. An aquapore is a protein that has 4 subunits. And you can see the 4 subunits being built up here. Those 4 subunits, each one has a channel for water. You can see the little holes here, and then there's a channel in the middle. We're going to take a little tour of one of the channels here. I'm going to kind of dive inside the molecule here and kind of be a water molecule. It's narrow, but the water molecules are going to fit easily through that little channel in the aquapore. There's the water as the little blue circles there. So we've just been talking about carbon dioxide and oxygen going through a cell membrane so far, and those can diffuse through without a pore, but almost everything else needs some kind of pore channel to go through the cell membrane. Water uses an aquapore, and other ions or molecules use different pores. So we've looked at an aquapore here, and let's look at a model that uses an aquapore. So first of all, let's put a mix of everything, different kinds of molecules, positive ions, negative ions, small nonpolars, that's the purple ones, small polar molecules, that would be water, medium polar molecules, sugar, and so we have all of those on a mix of everything. Right now, only those small nonpolar molecules, only carbon dioxide is getting through, can kind of slide through the cell membrane without a pore. We need to add an aquapore, a protein, remember those 4 protein sections that are coming together to make a channel in the membrane in order for water to get through. So now we can answer these questions. Which are the only types of molecules that pass easily through the cell membrane without an aquapore? And you can see what's up here besides blue. Look for the purple molecule in the key here. Also, let's ask what is true of most naturally occurring pores. They allow only ions to pass through. They allow most molecules. They let anything. They're specific to one kind of molecule. So at this point, you should be able to answer question 15 on your lab sheet. You've got some screenshots here to help you out. And question 16. I'll give you a hint on question 16. Read the highlighted stuff right here in purple. So now it's time to talk about osmosis. And osmosis sounds complicated, but it's really just a special term for the diffusion of water. Osmosis means the diffusion of just water. It doesn't mean anything else is diffusing, just water. And since all life requires water, being able to regulate how much water moves into or out of a cell is crucial. We have to figure out what affects the flow of water in and out of cells. So let's use a model to help us. So looking for those definitions at the beginning of your lab, here's a definition of osmosis. You can put it in your own words. Cells need to maintain a certain amount of water inside their membranes. Too much and they can explode, but too little and the cell's machinery will shut down. So we're going to use the model below to explore how the salt concentration inside and outside the cells affect the flow of water. So I'm going to make the salt concentration high on the outside. We'll make it low on the inside. And we'll run the model with a movable membrane here so you can see what's happening. And we'll look at the difference in osmotic pressure. It's a big difference at first, and there's a fairly big difference in the salt concentration at first. Water is going to diffuse to even out the concentration. The reason that the salt concentration is changing here is not because the salt is moving, it's because the water is moving to dilute the high salt concentration. Water is leaving the cell and going out here to kind of dilute the solution outside because it was a higher concentration. Now let's see if it runs in reverse. I'm going to reset everything. I'm going to make a low concentration on the outside and a high concentration on the inside, and we'll see what happens. Try to make a prediction for what's going to happen this time. This time, water, we know water is going into the cell. It's entering the cell because the cell is expanding. Water is coming into the cell to dilute the high salt concentration. It's going through the aquipores and diluting the concentrations, and it's trying to even everything out. As this evens out, the osmotic pressure will even out as well. So as the concentration changes, the osmotic pressure will change as well. When it gets to equilibrium, so these are equal, the pressure is equal, and the concentration is equal, you can see that the membrane is just kind of hovering where it is. As much water is going out as it is coming in. So let's go and look at the next question. Osmotic pressure is related to salt concentrations or other dissolved substances. In what way? High concentrations cause high osmotic pressure. Low concentrations cause high osmotic pressure. High concentration causes low osmotic pressure, or it depends on what's dissolved in the water. Let's stop this for a second and do a little bit of detective work here. So if we have none and high, and we it doesn't let us have any. We just have low and we have low. There's not much difference, and so there's not too much movement. If we stop and we reset one side to high, we're going to get a better osmotic pressure difference here, and because there's a bigger difference in salt concentrations. Remember, the difference in concentration is going to be related to the difference in pressure. So hopefully you can figure that one out. If you want water to flow out of a cell faster than into the cell, what should you do? Well, let's go find a situation where we can have that happening. We want water flowing out of the cell faster than into the cell. Here we've got a situation where water is leaving the cell is shrinking, and the water is flowing out of the cell faster than into the cell. So look at the concentrations here and figure out what you need to answer. Cells can generally stay in equilibrium with their surroundings. What are two ways you know that a cell has reached equilibrium? Well, let's look at what we were measuring and monitoring earlier. Here's one thing you can look at to look at if a cell is in equilibrium. Here's another thing. So that's a big hint for this question down here. Describe the similarities and differences between diffusion and osmosis. Well, diffusion includes osmosis because water is diffusing, but osmosis is not diffusion. Osmosis is diffusion of anything but water. If you're talking about osmosis, it's just the diffusion of water, just water. It's not the diffusion of carbon dioxide or oxygen. If you say osmosis, you mean just water is diffusing. So at this point, you should be able to answer question 17, question 18. There's some hints here in the screenshots. Question 19, also look at the screenshot, and question 20. So we're going to talk about making an electric potential. And the reason an electric potential is important to have across the cell membrane is a lot of different reasons, but one of the main ones is to send nerve signals. Frequently, cells need to keep high concentrations of some chemical inside, or they need to keep the chemicals out, having a low concentration inside. What happens when you actively move ions in or out of a cell? Well, let's find out. Through the process of osmosis and diffusion, nature tends towards a state where concentrations of everything are equalized. However, there are times when it's necessary to create a situation where concentrations are not equal. Concentration differences between the inside and outside of cells help to power the electrical signals in your nerves, give you strength to flex your muscles, and allows your cells to control the flow of water through osmosis. Below is a model showing a cell membrane, which you have seen is not permeable to ions. Try moving ions from one side of the membrane to the other. Do this until you build up a significant difference in the concentrations inside and outside of the cell and run the model. Can you think of a different way to try this out? Well, first of all, I'm going to hide the water molecules. I'll put them back later. I'm going to stop the model because it's easier to grab. And I'm going to build up a high concentration outside here. We know that the membrane is not generally permeable to ions, so we're pretending there's a pore in here, and we're using energy to move. I'm using my energy to move my mouse to move these ions physically across the membrane, just as an ion pump would. An ion pump uses energy as well. Using energy to move something across a membrane is called active transport. Now, if we run it, we can see that the outside concentration is different. The inside concentrations are different. And the electric potential is starting to develop. If we go back to the beginning, and we reset everything, watch the electric potential and watch these concentrations. Everything's even now and there's no electric potential. But when we set it up, when we move ions, and I'm moving ions here by active transport, I'm using energy to move these ions across the membrane through a pore. Now I'll run it. You can see the concentration differences occur and we get a voltage potential. So what must be done to get an electric potential? Well, I stopped and I moved some ions, so there was a difference in the concentration of negative ions on both sides, but the positive ions were pretty similar. Now, if I wanted to max out the electrical potential, I want to max out the differences. So I don't want positives to be the same anymore. Let's build up a positive charge on this side, because it's the difference in charge that really gives you a lot of electric potential. A lot of people explain voltage as kind of like a waterfall. Water at the top of a really tall waterfall has a lot of electrical potential, and just because there's a huge difference of where the water is and where it's going to be when it goes over the falls. And so the taller the falls, the more voltage. And you can bring that analogy here too. The more difference there is, the more voltage. So if you have a lot of positive over here and lots of negative over here, you're going to have a big difference. Let's see what happens to the electrical potential. Oh wow, lots more. When we get even more separation, we have almost all positive and almost all negative. We're going to max this out. So now you can answer these two questions. So at this point, you should be able to answer question 21 and question 22 on your lab sheet. So, what's the source of energy for actively transporting ions against their equilibrium concentration? So we know that that substances, ions and molecules, tend to move from areas of high concentration down to low concentration through diffusion. That's where they naturally go. But if you want to go up the concentration gradient from low concentration to high concentration gradient, you're going to have to use some energy. We created electrical potential by physically moving ions with our mouse from one side of the cell membrane to the other. A real cell needs some other source of energy because they don't have a mouse to make that happen. Adenosine triphosphate is a chemical found in all living things, provides the energy. When energy is used to move molecules across a membrane, it's called active transport. So when you're using ATP, that's called active transport. So I'll say that again because it's a definition at the beginning of your worksheet. When you're using energy to go push a molecule or an ion against the concentration gradient, in other words, from going low concentration to high concentration, to go from low to high concentration, you need some energy, and that energy is ATP. That process of going against the concentration gradient, against what diffusion would do, going from low to high concentration is called active transport because you need energy, you need some ATP. And we're going to watch that happen here. So watch the chemical energy, that's ATP, and watch the electric potential here. I'm going to set the ATP concentration to high. I'm going to set the calcium concentration to high. And what's going to happen here is we have a lot of calcium molecules. Stop this for a second. You can see it's a little bit lower. The concentration here, let's scroll down a little bit. They're even right now, but as it runs, this calcium concentration is going to change. The chemical energy is also going to change. What's happening here is in order to get across the membrane, calcium can't go by itself. It needs to use this pump. And what happens to the pump here is it waits for a calcium molecule, just kind of bumble in there. The phosphates aren't going to do. It's especially shaped that calcium fits in there. And it's also charged. And then it waits for an ATP to bumble by. So it has some energy to pump the calcium across the membrane. So when ATP kind of bumbles into this part, it's going to transform from ATP to ADP, and then it'll pop open the other way and push calcium across the membrane. So while we're waiting for the ATP to have a reaction here, we can talk more about here. As we use up ATP, we already used one, so you can see that the chemical energy went down a little bit and the electrical potential went up a little bit. So, what factor mostly affects how much chemical energy you start with? Well, the only source of chemical energy here is ATP. So, if we have low ATP, we're going to have low chemical energy. I set the ATP concentration here to high, so the chemical energy is fairly high here. Okay, I've let the model run a little bit longer, and you can see there's a little bit less ATP here. There's more just phosphate groups floating around, and that the chemical energy has gone down. In the meantime, as the chemical energy has come down, the voltage potential has come up. So every time we use up an ATP, this goes down a little bit, and every time we pump across a calcium molecule using that ATP, we increase the electric potential. So hopefully an ATP will bind here, get used up. There we go. There's the calcium, and you can see that the chemical energy went down because we used ATP, and the electric potential, because there's more difference in the calcium concentrations here, this electric potential happens because of the difference in the calcium concentrations. That's gone up as the chemical energy has gone down. So we haven't lost any energy, we've just transformed our chemical energy into electric potential. Now our next question here is describe how the chemical energy in ATP is converted into electrical potential energy, What we did, or what we're doing here, is increasing the difference in the calcium ions, just like we did in the last one where I physically moved the ions across the membrane to increase the potential. The calcium pump is doing it here, and instead of using the energy of pushing my mouse, which isn't a reality in a cell, it's using ATP to go ahead and move those calcium molecules and increase the difference in concentration, which sets up an electric potential. So I've put a number of screenshots here to help you out and give you some references here so you can answer question number 23 and question number 24, which we just talked about. The rest of the questions in the lab are questions you should be able to answer without any help from the model. There are no extra animations or anything that's going to help you out, and you can't check your answers even in the model. These are designed to be kind of a little quiz or assessment at the end of the lab. So I'm not going to help you with these questions. I'm going to let you think about it. You can go back in the video and look back at some of the models if you need to, and I'm going to let you think about these questions. Make sure you answer them or you won't get credit. Thanks so much. I'll see you in class. This is Miss R.